I often talk about how I view cancer as an evolving ecological system and how I find that this framework provides a lot of very interesting ideas and insights. This quite literally was the topic of my PhD. Today, I want to actually go through what that means and why I find this to be so interesting and possibly also very useful. So what does it mean, cancer as an ecosystem? Well, the idea here is that there are certain ecological features that are shared between what we think of classical ecosystems and tumors. For example, you can have predator-prey relationships like cancer-immune interactions, resources and competition for them, niche construction and environment modifications, migration and migratory patterns like metastatic seeding, unintended consequences of interventions. All of these things are observed both in tumors and in classical ecosystems. And the main question here really is not whether this is the right analogy or the wrong analogy or anything here. The question is it just interesting or could this actually be helpful? And I'm hoping to make a case that it is both. So let's start with predator-prey. This is really the most common relationship to conceptualize interactions between cancer and immune cells. And when we talk about that, what are the common features of predator-prey relationships? You would have prey that can grow without the predator. Okay, cancer cells would grow unrestricted without the immune system. The predator can kill the prey. Okay, that is obvious. Cytotoxic immune cells can kill cancer cells when they recognize them. Presence of the prey can increase the population of the predator. Okay, immune expansion. The predator, in this case, would be tumor-specific T cells, would die without the prey. We're not really talking about memory cells and a lot of those complexities, but in general, okay, so you have these features of the predator-prey system that could apply potentially to tumor-immune interactions. Like, this is not a perfect analogy, but that is not really the point. The point is it good enough to get us some possible utility? And I would argue that yes. And what I personally get out of thinking about tumor immune interactions as predator prey is looking for predator evasion strategies in other ecosystems and then think, do we see something like this in cancer? We can think of ideas like camouflage. Okay, maybe cancer cells look really like normal cells, so they're not recognized by the immune system. We definitely have that. Maybe some mimicry. That's a term for when one organism resembles another to gain evolutionary advantage. Okay, CD47, for example, could be the do not eat me signals, like the normal cells. Do not touch me, the immune system. Tumors obviously imitate chronic infections or wounds can imitate those to evade the immune system. We do have evidence of that happening. The tumors as wounds that do not heal. Maybe there's some interference with recognition mechanisms like incapacitating antigen-presenting cells or benefiting, like augmenting the predator's predator, the Tregs, or predator exhaustion mechanisms. In all of these interfering with the ability of the predator to do its job. So we definitely see a lot of these in the tumor immune interactions. And this framework really gives an idea to think about what other strategies do we know for predator evasion in other ecosystems. Can we search for analogous ones in cancer? These are just some of the examples, but maybe we can find more. So the idea here is what other ideas can we get if we think about it from this framework? Consumer resource relationship, that is another obviously very common and very important relationship in all the ecosystems. Cancer cells can be viewed as consumers that require resources. Okay, so you could have competition between cancer and immune cells for shared resources. There's some evidence of that happening. And the idea is if both cancer and immune cells can compete for, say, glucose, which both of them need, well, whoever gets to it first wins. One can think about then glucose upregulation, the glucose transport upregulations, and all of these mechanisms, not just as a survival mechanism, but maybe there is something that is additionally incapacitating the predator, something to think about and explore. Or intratumoral competition for resources. The idea here is that cancer cells compete with each other for resources within the shared carrying capacity of the organ, of the tumor. Well, this idea is actually the basis of the concept of adaptive therapy, which is really advanced particularly at Moffitt Cancer Center in Florida, both from a theoretical and also practical point of view. And the idea here is that you give therapy in such a way as to maintain the competition between therapy-sensitive and resistant cells within the shared carrying capacity to delay emergence of resistance. So we know MTD, you knock out all of your sensitive cells, only resistant ones remain. Well, can you give therapy in a different way, in such a way as you keep them interacting with each other so you do not have as much resistance? That's the idea behind adaptive therapy. Nutrient manipulation. Well, we know, for example, the examples of biological stoichiometry and phosphorus, like algae blooms. When you dump a lot of phosphorus into lakes, you get algae blooms. Phosphorus can be used for building DNA, so provides a lot of construction materials there. Okay, well, that gave rise to, for example, growth rate hypothesis. And the idea here was that adding excess phosphorus to the diet could shift this carbon to phosphorus ratio to favor highly proliferative cells. And this was actually experimentally shown in this experiment from 2008. They fed the mice, well, normal diet and high in phosphorus diet. And boy, did they see a difference in tumor diameter between the animals who were given normal chow and also the animals who were given high phosphorus diet. Does this mean that, tada, we have cure for cancer? Obviously not. But maybe this is one more puzzle piece, maybe one more little knob to tweak when we think about cancer more systemically. Another way to look at this is through the lens of limiting resource and Liebig's law of the minimum. Maybe different cells grow better with different resources, like hormone-dependent tumors. Well, we know that we have maybe somewhat better therapies for hormone-sensitive cancers. So the question can be, can we steer tumor composition to something that is more targetable through nutrient manipulation? Like pulling at different levers, not just pharmacological levers, but actually environmental levers, microenvironmental levers. We did theoretical analysis of this in this paper with Joe Brown. It was very fun. And then this year, this very, very interesting case study came out about restoring therapy sensitivity, particularly for prostate cancer, through very carefully timed testosterone injections. And this was really remarkable. So this is working with individual patients' data. So this is tailored to a particular patient. But the idea here was they developed resistance to androgen deprivation therapy, ADT, but by giving the carefully timed testosterone injections that favored the expansion of testosterone-dependent cells in there, and eventually they were able to restore sensitivity to ADT, which was absolutely remarkable. And as a side note, if anybody wants to work on things like that, personalized therapy like that, please reach out. That would be so interesting. But to the main topic, one can now ask the questions, what other examples have been observed in nature of nutrient-based manipulation and like evolutionary steering? What other strategies can we think of specifically for realistic nutrient manipulation? Some nutrients you can manipulate, some you cannot. Glucose, only to some extent, but other things you have much more flexibility with. Okay, now environment modification. A very classic example that we all know about, niche construction, process by which an organism alters its own local environment. And of course, when we hear this, we think beavers and dams. And this gives segue for me to talk about my favorite story of 2025 about Czech beavers. And if you haven't read about it, please do, because it's very joyful. In Czech Republic, they were trying to build, there was this giant project to build a dam. And then earlier this year, a colony of, I think, 8 animals basically did the whole thing overnight. And I find this to be very wonderful and remarkable. But do we see niche construction in cancer? Well, yes. A classic example would be acidic microenvironment that happens as a byproduct of metabolism, particularly of increased glycolysis. What are the consequences? Well, competition. Maybe normal cells are less able to deal with low pH. And we do know that, for example, some transporters are upregulated on cancer cells to basically pump out more acid. Interfering with a predator, and it can, like low pH, can affect immune cell priming and maturation. Again, we can think about it evolutionarily, why this would have evolved. I would argue that this is actually a normal adaptation, like in many cases during wound healing, but that's a different topic. Environment that's more, this could be also acidic microenvironment, could be more permissive of migration. Again, maybe weaker membranes, easier for cells to move. What do we think about that? Another example of environment modification is angiogenesis and new blood vessel formation. What does that actually mean? Well, practically, this can mean that the tumor is essentially increasing its own carrying capacity. So this is an idea of dynamic carrying capacity. It could facilitate migration. There are more open roads, basically, and so facilitate metastatic seeding, for example. It can also increase access to nutrients for them to go to the tumor, but it also increases access to drugs. So you can talk about now the payoffs. You're choking angiogenesis. Yes, maybe you're starting to starve some of these things out, but you're also no longer able to get the drugs in there. Things to think about when we're balancing these. This is not all very straightforward. And so, but these are just, again, examples of environment modification to start the discussion. What other examples of niche construction and environment modification do we see in other systems? Is it something that we can, now that we know these things, maybe these ideas exist, can we now see them in tumors? Maybe that can give us some new ideas that we didn't think of before. Oh, and of course, are these adaptations reversible? Because that is another question that could also be explored. Migration and migratory patterns. Well, an obvious analogy is thinking about metastases as migrations. Questions: how and why cancer cells leave the primary tumor? There is a lot of theory around that. Maybe going towards something good versus going away from something bad. Different strategies can be thought of based on these different hypotheses. Can you, quote unquote, lure them out? What is chasing them away? These ideas. The question of how they travel. It seems like they travel in clusters. Okay. Why clusters? Why is it more successful? Currently, we think of metastatic cell travel through plumbing, like blood vessels and lymphatic systems. Are there other possibilities? Is there anything, the idea of metastatic niches, environments that are more welcoming to metastatic clusters? Can we find any of these analogies in other ecological systems? And is there something that can be thought of here? And of course, how they settle new environments. As I said, metastatic niches. These are all very interesting and exciting questions, and maybe we can get additional insights if we think of them as relationships in other systems. What migratory patterns can be observed? Can they be influenced? Can they give ideas for any of the questions above, so to actually maybe do something? Another topic that, of course, happens is unintended consequences of an intervention. So here, this was just a very cool example that I saw in the literature, and I just want to share on this. A group of wolves from Jasper National Park in Alberta arrived in Yellowstone National Park on January 12, 1995. That changed the population size and behavior of elk. Well, so now the elk were hiding more, not grazing as much. That allowed more trees to grow to maturity because the little elk were eating the saplings. That in turn led to recovery of aspens and other trees. So you have this trophic cascade, this chain reaction of effects from wolves impacting the elk impacting plants or not taking the plants can now grow to maturity, and so wolves allowed for aspen trees to regrow in the park. I find that to be both a wonderful example, but also an example of this indirect impact of interventions, that you have these chain effects that we don't often think through in advance, and maybe we should. My favorite recent example of unintended consequences in a cancer system has been, I've talked so much about this, but I really, really love this, the PI3K inhibitors. This was a paper by Hopkins et al. From 2018. And essentially, so the authors, they gave mice a tumor and then treated them with PI3K inhibitors. They saw response, great. But just because of the mechanism of action, PI3K inhibitors, they affect metabolism. So that resulted in the mice actually developing transient diabetes. So glucose went up and insulin went up. Insulin can feed tumors. So now, yes, you have maybe some negative effects on tumor growth from the drug, but you have positive effects on tumor growth from insulin, which was indirectly caused by the drug. And so we saw tumor regrowth. Well, what they did then is they treated mice for diabetes. They did not try to do other treatments. They actually tried to reverse this particular adaptation. They did metformin, they did SGLT2s, and also keto diet. Once they normalized blood sugar, and more importantly, normalized insulin, they restored the response. I find this to be a fascinating and amazing example of when an adaptation triggered by the drug, but it's unrelated to its intended mechanism, resulted in loss of efficacy. And it's reversing this adaptation that actually allowed resensitization to treatment. And the idea here is not only about, of course, multiple unintended consequences, but also one could have approached this by, oh, let's add more drugs to this regimen or do this, do that. But instead, actually looking into this and normalizing specifically, I'm saying specifically insulin, it was not about glucose, that allowed restoring sensitivity. I did a model of this because I went down a rabbit hole on trying to capture this emergence of insulin resistance and how it can affect cancer. So a very conceptual idea, but nevertheless, if you want to take a look, it's out. Okay, but more broadly, what examples of unintended consequences have we observed in other cases? What natural experiments can we see? Can we inform any strategies of that? From that, this is something to think about, unintended consequences of interventions. And in general, like fishing ideas from different fields to have this more systemic and holistic thinking about ecological thinking about cancer. Like, for example, conservation biology. Well, habitat modification. To save species, preserve their habitat. We know that. If we want to do the opposite, if you think of cancer as a species that you want to eliminate, what parts of its habitat, of its microenvironment can we modify? Pest management strategies. If we can't get rid of them, how do we coexist with them? How do we manage? So the idea here is with keeping pest populations at a manageable level can actually help prevent emergence of resistance. Maybe something in extinction theory. Last time I looked at it was the press pulse hypothesis from Ahrens and West. I do not know if there's maybe new insight on mass extinction events, but nevertheless, something to look at. What if we want this species, if you think again of cancer as a species within the human body, what if you want them to go extinct? Can we learn any strategies from mass extinction events? And in general, I find really natural experiments to be a very fruitful way to think about these things. And why is it so important to think systemically in interventions, particularly with a really complex disease of cancer? Well, this is how I personally think about it, is that in designing treatments, we tend to focus on the big contributors to efficacy, like chemo and immunotherapy. We're not just trying to kill the cancer cells directly. We're at least now augmenting its natural predators, so stepping a little bit out. That's already good. But since it's a system, there may be additional knobs to get us to the outcome that we want that are currently not being utilized. This is kind of a mathy analogy, but if you like it, I like to think of it as Taylor series. You have linear terms that contribute to most of things, but then you have higher order, a little bit of here, a little bit of there to tip over. Another example that I like, this is a term I learned from Peter Attia, majoring in the minors versus minoring in the majors. So focusing a lot on the minor things, like, I don't know, phosphorus, versus not enough on some big things. So how do I see this? These are made up numbers, but I just wanted to show an idea. So for example, we do chemo that gets us 35% there. And maybe we add immunotherapy and that gets us maybe 45% there. And maybe we get a little bit more from glycemic control. And maybe we get some more from nutrient manipulation. And maybe we get an extra 4% from increasing muscle mass, which has been actually shown to be very, very important in this case as well. But what else? So yes, little bits of pieces. We tend to think that if we get these guys, then we got there. But that's not enough. And I would argue that these things, yes, they may be little contribution a little bit here, a little bit there, a little bit there, but maybe there is something into thinking about how we can have a more holistic approach here. Why are these things not utilized? Why are they actually looked at as really not important enough? Well, I took these pictures when I was walking around Crane Beach in Ipswich a few years ago. And this is how, again, I personally think about this. Well, maybe you can have a simple disease where you can have, well, maybe one thing goes bad here and maybe here and here. And then you pinch one of these or the other one, and pinch these streams, and then you kind of stop what goes downstream here. A complex disease will have many, 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 many more streams going there. Just pinching one or the other, one or two or three, is not going to get you there. So one or two interventions will not get you there in a complex system. You have to figure out much more multi-pronged approach here. And I would argue, and I believe cancer is closer to this, but we keep treating it as if it's this. And that is not the right way to do it. Why do we keep doing it? Well, scientifically, we can and should be testing one thing at a time, and that makes a lot of sense. But this approach is not designed for interventions in a system with so many possible levers and unintended consequences. So this is something that really needs to be thought through. How do we adapt to this disease being different? Clinical trials are also designed relative to standard of care. And again, if you think about it from the point of view of clinical trial design and just being ethically responsible, it is the conservative approach to both design and evaluation of trials. It is very conservative and it is ethically appropriate, but it does make it hard to test alternatives outside of critical situations like terminal cancers, pediatric cancers, things like that. How do we think about this? I keep thinking about the initial success of immunotherapy being contingent on basically one person pushing for different trial endpoints for immunotherapy compared to what we do in chemo. It should be easier to do that. Finally, this is where I will make a case for us as modelers really contributing here, because as modelers, we can conceivably build a stronger case for systemic interventions. We can simulate individual interventions and systemic treatments and put them together. And we can really make a case for this quantitatively driven change in protocols that is not a guess, that is driven by data and mechanism and understanding. And we can play with all of these levers, or test. Play is probably not the right word here. We can test these levers in an in silico system before we take it forward. So a couple of other thoughts on mathematical modeling of cancer. One challenge here is, and in biology in general, is that there is no right model. You can hear a lot of words about big model, small model, this model. There is no right model. We are not physics. There's model for the data, there's model for the question, and I find that probably is very important to not get too hung up on specific functional forms of equations or levels of detail. Levels of detail should be appropriate for the question and for the data. But it also is liberating that there is no right answer, one right answer. As a colleague of mine used to say, when we would approach the same question or the same decision from different points of view, there are multiple ways to climb a mountain. And there's liberty in that. Also, in biology, nothing is definitive. Everything we can predict is only really in terms of probabilities, including us as modelers, and the best we can try to do is really increase the likelihood of a desired outcome. Yes, we can have deterministic models, but of course we all understand the limitations of them, and we are working on tools and methods to incorporate heterogeneity, virtual populations, uncertainty quantification, all of these things to really increase the likelihood of desired outcome. Mathematical modeling is not magic, but it is a wonderful and remarkable tool. Okay, final thoughts on this topic, because I did not have a specific bucket for them, but I really feel like they're important to mention. Efficacy versus effectiveness. So efficacy, if it works in perfect conditions with perfect compliance. Effectiveness, if it works in the real world. Classic example, diet and exercise. Very effective, but not very efficacious, but not effective. And we should plan and simulate both, particularly for effectiveness, for compliance, for any variability in individual characteristics. Natural experiments, I find that to be a fantastic source of ideas and information and hypotheses to look through. And key part of interventions in a system, both intended and unintended consequences. And I find that it's really important to anticipate both, to plan for it, like planning for resistance as opposed to being surprised by it every single time. We are looking at we're currently developing some methods on anticipating resistance. Stay tuned to how that turns out. If you want to talk about applying it to your data, please reach out. But in general, anticipating things and planning for them is, I find, to be much better strategy than being surprised. Okay, some references. This was the experiment with the dietary phosphorus. This was the amazing paper on evolutionary steering in prostate cancer with testosterone injections. This was my favorite experiment for several years, as I was thinking as to how to describe it mathematically, or at least the idea of emergence of insulin resistance and diabetes. This was an experiment with drug-inducing transient diabetes when treating it restored drug sensitivity. This was just my personal paper on insulin resistance and cancer. Liebig's law of the minimum applied to, again, this is a theoretical model, but can be expanded to actual data, obviously. So this was on estrogen. This is some work that we did on ideas of mitigating non-genetic resistance to checkpoint inhibition and thoughts about immune cell exhaustion. And this is just a fantastic book on the history of immunotherapy that I think everybody should read because it was really interesting. And that is all I got. Thank you so much for your attention, and please reach out if you want to talk more.